When we look at the chemical events that sustain life, we will see that most of these events occur inside cells. Much of the anatomy and physiology cannot be seen with the naked or unaided eye because cells are very small, and as a result, no one could actually examine the structure of a cell until effective microscopes were invented. There have been several significant events throughout history that have led to the development of the field of cell biology as it exists today. Let us look at some of the proponents and their contributions that led to the development of the cell theory. Robert Hooke is an English scientist who published the book Micrographia, which describes organisms that he viewed under the microscope of his own invention. While observing a cork, he noticed a box-shaped structure which he called cells because they reminded him of the cells or rooms in the monasteries. This discovery led to the development of the classical cell theory. However, at the time, Hooke was not aware that the quark cells were long dead and therefore lacked the internal structures found within living cells. Inspired by Robert Hooke's discovery, Anton van Leeuwenhoek, a Dutch merchant and amateur scientist, conducted his own experimentations and was probably the first to observe live microorganisms through the magnifying lenses of more than 400 microscopes he constructed. He wrote a series of letters to the Royal Society of London describing the animalcules he saw through his simple single-lens microscope. Leeuwenhoek made detailed drawings of animalcules he found in rainwater, in his own feces, and in material scraped from his teeth. These drawings have since been identified as representations of bacteria and protozoa. Because of his discovery, Leeuwenhoek is universally acknowledged as the father of microbiology. Matthias Schleiden, a German botanist who made extensive microscopic observations of plant tissues, described them as being composed of cells. Theodor Schwann, a noted German physiologist, made similar microscopic observations of animal tissue. In 1839, after a conversation with Schleiden, Schwann realized that similarities existed between plant and animal tissues. This laid the foundation for the idea that cells are the fundamental components of plants and animals. Because of these observations, Theodor Schwann proposed the classical cell theory. Together with Schleiden and based on their observations of the plant and animal cell, they concluded that all organisms are made up of cells and that cells are the basic unit of life. Robert Remak, a Polish scientist and a prominent neurologist and embryologist, published convincing evidence that cells are derived from other cells as a result of cell division. 3 years later, Rudolf Virchow, a well-respected pathologist, published an editorial essay entitled Cellular Pathology, which popularized the concept of cell theory using the Latin phrase omnis cellula e cellula, meaning all cells arise from cells, or that cells come from pre-existing cells that have multiplied. Since the formation of the classical theory, technology has improved, allowing for the more detailed observations that led to new discoveries about cells. These findings led to the formation of the modern cell theory, adding 3 more tenets to the fundamental tenets of the classical theory. These tenets state that DNA is passed between cells during cell division. Cells within similar species are mostly the same both structurally and chemically, and that energy flow occurs within cells. Despite their complexity and variety, all living cells can be classified into two groups based on certain structural and functional characteristics. Prokaryotes and eukaryotes are chemically similar in the sense that they both contain nucleic acids, proteins, lipids, and carbohydrates. They use the same kinds of chemical reactions to metabolize food, build proteins, and store energy. It is primarily the structure of cell walls and membranes and the absence of organelles that distinguish prokaryotes from eukaryotes. The chief distinguishing characteristics of prokaryotes is that, first, their DNA is not enclosed within a membrane, and it is usually a singular circularly arranged chromosome. Some bacteria, like the Vibrio cholerae, have 2 chromosomes, and some bacteria have a linearly arranged chromosome, but most of the time it is a singular circularly arranged chromosome. Second is that their DNA is not associated with histones. Histones are special chromosomal proteins that are found in eukaryotes. There are other proteins associated with the DNA but not histones. Third is that prokaryotes lack membrane-enclosed organelles and that their cell walls almost always contain the complex polysaccharide peptidoglycan. And then lastly, they usually divide by binary fission. So during this process, the DNA is copied and the cell splits into 2 cells. Binary fission involves fewer structures and processes than eukaryotic cell division. As compared from prokaryotes, eukaryotes' DNA is found in the cell's nucleus, which is separated from the cytoplasm by a nuclear membrane, and the DNA is found in multiple chromosomes. In addition, their DNA is consistently associated with chromosomal proteins called histones and with non-histones as well. And they also have a number of membrane-enclosed organelles, including the mitochondria, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and sometimes chloroplasts. And their cell walls, when present, are chemically simple. Cell division in eukaryotes usually involves mitosis, in which chromosomes replicate and an identical set is distributed into each of 2 nuclei. This process is guided by the mitotic spindle, and the division of the cytoplasm and other organelles follows so that the 2 cells produced are identical to each other. In this video, we will focus on specifically human eukaryotic cell. And there are 3 major parts of the cell, and that is the plasma membrane, the cytoplasm, and the nucleus. So, the plasma membrane forms the cell's flexible outer surface, separating the cell's internal environment from the external environment, and it is a selective barrier that regulates the flow of materials into and out of a cell, and this selectivity helps establish and maintain the appropriate environment for normal cellular activities. The plasma membrane also plays a key role in communication among cells and between cells and their external environment. The cytoplasm consists of all the cellular contents between the plasma membrane and the nucleus, and this compartment has 2 components. It has the cytosol and the organelles. The cytosol is the fluid portion of cytoplasm, and it contains water, dissolved solutes, and suspended particles, and within the cytosol are several different types of organelles. Each type of organelle has a characteristic shape and specific functions. Example of these organelles include the cytoskeleton, the ribosomes, the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and the mitochondria. The nucleus is a large organelle that houses most of a cell's DNA, and within the nucleus, each chromosome contains thousands of hereditary units called genes that control most aspects of cellular structure and function. Now, when we say chromosome, this is a single molecule of DNA associated with several proteins. The different substances that make up the cell are collectively called protoplasm. So, protoplasm is composed mainly of 5 basic substances, and that is water, electrolytes, proteins, lipids, and carbohydrates. Most cells, except for fat cells, are comprised mainly of water in a concentration of 70 to 85%. Many cellular chemicals are dissolved in water and others are suspended in the water as solid particulates. So, chemical reactions will take place among the dissolved chemicals or at the surfaces of the suspended particles or membranes. Now, there are also important ions in the cell, which includes potassium, magnesium, phosphate, sulfate, bicarbonate, and smaller quantities of sodium, chloride, and calcium. The ions provide inorganic chemicals for cellular reactions, and they are necessary for the operation of some cellular control mechanisms. And example of these are ions acting at the cell membrane that are required for the transmission of electrochemical impulses in nerve and in muscle fibers. After water, the most abundant substances in most cells are proteins, which normally constitute 10% to 20% of the cell mass. These proteins can be divided into 2 types, the structural proteins and the functional proteins. So, structural proteins are present in the cell mainly in the form of long filaments that are polymers of many individual protein molecules. A prominent use of such intracellular filaments is to form microtubules which provide the cytoskeletons of cellular organelles such as the cilia, the nerve axons, the mitotic spindles of cells undergoing mitosis. Functional proteins, on the other hand, are usually composed of combinations of a few molecules in tubular-globular form, and these proteins are mainly enzymes of the cell, and they are often mobile in the cell fluid. Also, many of them are adherent to membranous structures inside the cell, and they catalyze specific intracellular chemical reactions. Another component of protoplasm is lipid, and lipids are several types of substances that are grouped together because of their common property of being soluble in fat solvents. Especially important lipids are phospholipids and cholesterol, which together constitute only about 2% of the total cell mass. Phospholipids and cholesterol are mainly insoluble in water and therefore are used to form the cell membrane and intracellular membrane barriers that separate the different cell compartments. In addition to phospholipids and cholesterol, some cells contain large quantities of triglycerides, which is also called neutral fats, and in fat cells or adipocytes, triglycerides often account for as much as 95% of the cell mass. Now, the fat stored in these cells represents the body's main storehouse of energy-giving nutrients that can later be used to provide energy wherever it is needed in the body. Carbohydrates play a major role in cell nutrition and have structural functions as parts of glycoprotein molecules. Most human cells do not maintain large stores of carbohydrates, and the amount usually averages only about 1% of their total mass, but it can increase to as much as 3% in muscle cells and occasionally to 6% in liver cells. However, carbohydrate in the form of dissolved glucose is always present in the surrounding extracellular fluid so that it is readily available to the cell. Also, a small amount of carbohydrate is stored in cells as glycogen, which is an insoluble polymer of glucose that can be depolymerized and used rapidly to supply the cell's energy needs. This is TinkPharma with KJ Navarro, and if you find this video helpful, don't forget to hit that like and subscribe button and to activate the notification bell so you can get updates on my latest videos.